I'm an adult nephrologist currently based in Switzerland, Lausanne, after spending a lot of years in France, and I have for several years an interest in all complement-mediated rare disease. And I coined the term C3 glomerulopathy almost 10 years ago. And I have been involved in the development of several complement inhibitors in atypical HOS, but also in C3 glomerulopathy, immune complex MPGN, and other complement-mediated kidney disease. They are rare diseases, but they are so severe that they have attracted a lot of attention because until the availability of the new complement inhibitors, they had no efficacious, proven, specific treatment. So, C3G is a disease where you have complement degradation product, meaning C3, that goes into the kidney and alters the function of the kidney. And it results from an excessive activation of a key enzyme of the complement system called alternative C3 convertase. So it is overactive, and this leads to the deposition in the kidney of a huge amount of C3 degradation product that leads to the damage. And this translates in some morphological changes in the glomerulus that we can term sometimes membranoproliferative changes, thickening of the basement membrane, and some proliferation and expansion of the mesangial membrane. And you have another disease which is very close, called immune complex MPGN, where you have the same process with the deposition of both complement degradation product and immunoglobulin. And to date, we are not sure if they are completely different, or whether they are two aspects of the main disease seen at different times of the course of disease in a given patient. But in all cases, in both diseases, you have signs and features of complement alternative pathway activation, including in immune complex MPGN. So they are severe because you have roughly 50-16% risk of reaching end-stage kidney disease. And they have a double burden on patients because they recur very frequently in the kidney transplant, up to 50%, and they lead to the loss of the kidney transplant due to the recurrence of the disease, despite all the treatment given as immunosuppressor for the renal transplantation. So, very rare disease, but more frequently recognized, no specific treatment, no efficacious treatment approved, and the high risk of recurrence after renal transplantation. This is a global picture. It is part of the innate immunity. This is the first line of protection against infection. It's a very old system with three pathways, roughly. You have the classical and the lectin pathways that are activated by immune complexes and necrotic bodies. And you have the alternative pathway that is triggered by infection, and it is in the low state of activation due to the fact that it needs to be rapidly amplified if you get a meningococcal infection or a pneumococcal infection. Roughly, the alternative pathway that helps people to get to the hospital alive when they have a meningococcal meningitis or pneumococcal pneumonia. This is the first line of defense till you get the antibiotic. And we have seen more and more frequently identified acquired or genetic causes of dysregulation of this alternative pathway. Alternative pathway, as it is in a low state of activation, it is tightly controlled. So some patients lose the control of this activation, and they have an excessive activation of the alternative pathway, leading in some cases to the deposition in the kidney of a huge amount of C3 degradation product. So the starting point, as I said, is that we had no treatment for approved efficacious treatment for C3G and immune complex MPGN. What we usually use as first-line therapy is mycophenolate, mofetil, and corticosteroids that works in some patients and usually very transient. And when they recur, the disease recurs after renal transplantation, usually patients are already on MMF and corticosteroids. So you have already used the first-line treatment, and it didn't prevent the recurrence in a significant proportion of patients. So the idea is to target the C3 convertase activity directly by blocking C3 and C3b, which is one of the first steps of the activation of C3. So with Pegcetacoplan, you are targeting the main driver of the disease, which is C3 activation and release of degradation product. So this drug was tested in patients who had recurrent C3G or immune complex MPGN in the kidney transplant. It was prospective, randomized with a control group, small control group of 3 patients who had the standard of care. All of them had a rebiopsy at 3 months, and they were followed up till 1 year, 52 weeks. The primary endpoint was the reduction in the deposits of C3 in the kidney transplant on rebiopsy done during the follow-up, mainly at 6 months. So this was the main design. You had additional endpoints were inflammation in the kidney and proteinuria for those who had initially proteinuria, because not all of them had proteinuria. They were included based on pathological recurrence. The results were really very impressive in terms of reduction of C3 deposition. Roughly 80% of patients who went to 52 weeks of treatment had a reduction and complete disappearance of C3 deposits in the kidney. And this reversal of C3 deposits have not been seen till the availability of Pegcetacoplan and to some extent other complement inhibitors. This is a major finding because the aim of the treatment of C3G and immune complex MPGN is to reverse the C3 deposits that are harming the kidney. 80% achieve complete disappearance of C3, and this was associated to a decrease in inflammation in glomeruli as assessed by an inflammatory score, but also in reduction roughly by 45 to 45% of proteinuria in those who had proteinuria at baseline. And this was associated to a stabilization of eGFR. So the picture of efficacy in this limited number of patients still, but still very promising data and very impressive data because you have a succession of decrease in C3 deposits, decrease in inflammation, decrease in proteinuria, and stable eGFR, which gives a lot of hope for finally having available new specific efficacious treatment for recurrent C3G and immune complex MPGN in kidney transplant. The next, as you know, it is tested in a prospective control versus placebo trial in native kidneys and recurrent C3G and immune complex MPGN in a large trial called Valiant trial, which included a very impressive number of patients, more than 120 patients, which is a very significant number for such rare disease. So the trial is ongoing, and I'm very hopeful and I'm very quite certain that it will confirm the initial data in kidney transplant recurrent C3G, but also extend the efficacy, the proof of efficacy of Pegcetacoplan also native kidneys. And the primary endpoint in this Valiant prospective trial will be proteinuria, which is plus reduction in C3 and stabilization of eGFR and inflammatory change in the kidney. But the primary endpoint is proteinuria, and we are all eagerly waiting for the results.